welcome to the Dementia Doctor. Today I have Dr. Bhatt with me, and he is going to, first of all, introduce himself, and then we're going to talk about his research.
So my name is Nemil, and I'm a postdoctoral fellow at University of Texas Medical Branch under the mentorship of Dr. Khushboo. And what our lab essentially does is it designs novel therapeutics against Alzheimer's and other dementias. And that is kind of our bread and butter of the lab.
Okay, welcome to the channel. And we have a lot of our listeners who are dealing with Alzheimer's disease. So we have two proteins for Alzheimer's, that is amyloid and tau, that accumulate in this disease. Tell me more about which protein do you target and how are you targeting it?
So in the field, probably more than 100 years, we've known that there's amyloid beta is one of the proteins, and then later we found out that there was tau as well. And so most of the current therapies that are out in the market are all targeting amyloid beta, which is said to come first in the disease, and then tau ends up coming second, is what is believed, at least. And so our lab kind of targets the tau protein, which is believed to be the executioner of the disease, as opposed to amyloid beta.
Yeah, I would agree. In our clinical understanding also, we think that tau, although comes second after amyloid, it is closely related to clinical decline or memory changes, as well as shrinkage or atrophy we see in the brain. So tell us a little bit more about how are you targeting this protein? What are you doing?
So our approach, or the novelty of our approach, is that we have taken from patients who've passed away their brains, and we've isolated their tau, the diseased tau, and we've designed an antibody against it. And the novelty or the strengthened part about this is that it allows for us to target only the disease-relevant tau. So tau is a very important protein in the neuron. It serves to kind of, it's like the railway track almost. It supports the railway track of the neuron. And so if we target some of the physiologically relevant tau, the naive tau, the healthy tau, what ends up happening is you may get off-target effects. And what we did was we targeted only the diseased tau by isolating that disease tau and generating an antibody against it. We're more likely, or at least we're hopeful, that we can then bring this from preclinical into a clinical setting.
Yes, of course. I think the tau, as we know, is the backbone or railroad tracks or the skeleton of the cell, carries all the nutrients up and down the cell. So targeting the healthy tau would not be helpful for us, but targeting the diseased tau is what we are hoping for. And I think your therapy approach is also very interesting, because a lot of times we see individuals who are making therapies with the mouse models of the tau or other ways of targeting therapies. You are looking at human beings who have passed away, unfortunately, from Alzheimer's disease, and then removing or getting a hold of the tau protein from their brains and then developing a therapy against it. So how far are we in this development process? How close to the clinic are we?
So our goal is to get it to the patients as fast as possible, but we are still in the preclinical setting, and we're going through the appropriate channels to get our novel antibody approved by the Federal Drug Administration.
Okay. So for our listeners, most of the medications that we have go through preclinical setting or preclinical stage, and then we go into phase 1, phase 2, and phase 3 trials, and it can depend on how long it takes from a drug to move from one phase to another phase. But typically it's on a period of years at least to say. So we are in very early stages, but it's encouraging to see that you're doing this research. I also am very interested about the news that we heard about your lab, and we heard that you're developing an intranasal therapy?
Correct. So unfortunately, when we administer these anti-amyloid therapies, most of them are through the IV, which means that only a little bit actually goes to the target organ, which is the brain, right? And so what we said was there has to be a better route, and we exploited a physiological route that is present. And so we said if we can use the intranasal route through the nose, and if we can deliver it right to the brain, we can bypass the entire circulatory system and deliver more of the drug into our target.
Yeah, I do think it's a very novel idea, and it's something that will be good for individuals who fear needles. And of course, thinking from the perspective of biology, something that goes IV goes to all your organs, whether
Yes.
you want it to go or not. But something that is intranasal or a nasal spray has a more availability in the brain. So that again is interesting. So are you developing it against the amyloid protein or the tau protein?
So currently our approach is to develop it against the tau protein.
Okay.
But if successful, and we can use the same approach against a lot of different diseases. It's not
Yes.
just about Alzheimer's, it's also about FTD, PSP. There's a whole, what they call AD related and related dementias. So
Absolutely. Yeah, all the proteins that accumulate in the brain, and nose is a good pathway to the
Yeah.
brain, because we have a nerve of smell that comes from the brain directly to the nose, and so that is a good way to target the brain directly. So of course, if this is developed, it would target other proteins that cause other dementias in the field. My understanding is that this is also in the preclinical stage right now?
yes, ma'am.
Okay.
Yes, ma'am.
Okay.
And so we've compared both IV and intranasal, and hopefully our goal is to administer less of the drug intranasally compared to the IV method, and kind of compare both the methods side by side. So say if we administer twice the drug IV and half the dose intranasal, then we can get a better understanding of the efficacy and how important is the penetration of the drug into the brain.
Okay.
Yeah.
Now, I'm really interested in what does this preclinical stage of a drug approval looks like. Is it testing in animal models? What exactly are you doing?
So currently we're testing in mice is
Okay.
our primary go-to, but ideally we'd also like to move to other animal models as well, just so that we can clear the regulatory agencies'
Okay.
mandates.
And are these mice models the ones who developed tau, or?
Correct. So
Okay.
they all have been genetically made so that they develop an aspect of the tauopathy.
Okay,
Yeah.
okay. So these are, for our listeners, these are mice that are changed to make these proteins that are typically seen in Alzheimer's disease, the tau protein, and that is one way that our research moves forward. We have the mice that make that protein, and we can give the medications that can remove that protein and see how well they are doing the job of removing the protein that we are targeting.
Yep, that's very well put.
Anything else that you would like to share with our listeners?
Stay hopeful.
Okay.
There's a whole bunch of research going on trying to solve this one problem. So.
Yes, I mean, it is, although it's in the preclinical stage, we will see this coming in the next couple of years. So
Yeah.
it is most hopeful time in Alzheimer's disease. Thank you so much for joining us
Of course.
today.
Thank you for having me.